Hi everyone, my name's Robin Shaw and I'm really excited to share some of my research with you today about how I use genetics to learn about marsupial ecology. So I work with the Western Australia Department of Biodiversity, Conservation and Attractions, but I work for them remotely from Canberra, where I'm also a visiting researcher at the Australian National University. Before getting stuck into the research though, I wanted to acknowledge the many different traditional owner groups in the Pilbara region, which is an area that I've been working in over the last few years. I wanted to recognise their massive contribution to scientific knowledge and research, as well as their ongoing stewardship of country. I also wanted to acknowledge that I'm speaking to you from Ngunnawal and Ngambri land today and pay my respects to elders past and present from both regions, as well as the Whadjuk Noongar people. So I wanted to start by giving you a little bit of background about myself and the types of studies I've done in the past, as well as what working in research can look like, whether that be at universities, NGOs or conservation agencies. So I'm from Canberra and I did my undergrad through to PhD at the Australian National University. But for my PhD I was also lucky enough to partner with the Australian Wildlife Conservancy, and I did all of my fieldwork up at their Mornington Wildlife Sanctuary in the Kimberley. So it was definitely during this time that I absolutely fell in love with Western Australia, and in particular with these really vast remote landscapes like the savannas in Northern Australia or the arid zone. So the main question I was asking during my PhD was, how do animals recover after disturbance? And one really pervasive disturbance in Northern Australia is of course fire. So I trapped this very cute native rodent, the pale field rat, and carried out different experimental fires. So management fires, which are low intensity and patchy, and also some really high intensity thorough fires that mimicked wildfires that generally occur late in the dry season. And we wanted to know whether these different fire types promote different recovery mechanisms. And we used field observations as well as genetics to find this out. And I'll tell you a little bit more about how we can use genetics in this type of research later on. And so what I found was after patchy burns, we had a lot more animals surviving and then repopulating sites from within. So recovery can potentially happen much faster if you have unburnt patches over the scale of an individual's dispersal capacity. However, in these really thorough, intense fires, of course, animals find it really hard to persist after because there's no habitat to protect them from predators. But depending on the scale of the fire, recolonisation has to happen outside of that area, and so it can take many generations for animals to actually get back in if it's a really, really vast wildfire. And so all of this information can be incorporated into AWC's fire management strategies and is really important for, I guess, tailoring those management strategies towards biodiversity conservation. So after my PhD, I did a couple of short stints at different universities. I went up to Charles Darwin Uni and finished off some of this fire research. And then I went over to the UK to Cardiff University, which was a lot of fun. I got to see a lot of rugby and eat a lot of Welsh cakes, so it was a great time. But I also kept working on rodents. I'm sorry, marsupials will come in later on. But for this project, I was studying this very cute multi-mammate mouse, which is native to sub-Saharan Africa. But it's also a vector for disease for humans. And so we wanted to know what makes this mouse so good at colonizing new environments and whether we can predict areas that it might colonize in the future with climate change. And again, we use genetics to look for patterns of adaptation in this species. And then after Cardiff, I moved to Perth, where I did a postdoc with Murdoch University and also the Department of Biodiversity, Conservation and Attractions, or DBCA. And this was the project where I started working on small mammals in the Pilbara region. And of course, this one includes some marsupials, so I'll be telling you a lot about this work later on. During this time though, I was very lucky to get out with DBCA quite a bit and doing some fieldwork on booties or burrowing bettongs, and also black-flanked rock wallabies in the Little Sandy Desert. So I've really enjoyed all of these different projects for a nice mix of lots of fieldwork, really interesting scientific questions, but also the use of genetics and combining these different disciplines. And I guess the main focus of a lot of my research is using genetics as a tool to understand the ecology of animal populations, and a real focus on translating this information through to conservation. And so that's why it's been so great to work with DBCA and the Australian Wildlife Conservancy to figure out how we translate this research into something practical. And so now, as I said, I'm at the ANU based at the Australian National University, but still working with DBCA and Pilbara small mammals. So why do I use genetics in my research? Well, first of all, genetic diversity in itself is just a really important element of biodiversity. So it's one of the three main pillars of biodiversity, with the other two being species diversity and ecosystem diversity. But it's probably the foundation of these other two pillars because it underlies the health of a population. And so what I mean by this is that genetic diversity is the variation upon which natural selection acts. So if we think of this example where we have lots of different genetic mutations, so high genetic diversity, if we have some extreme event happen, it's more likely that because there's a lot of variation in the population, there might be a few individuals that actually carry a gene that is adapted to this new environment. And so you might see species persist because they have the genetic ability to do so. The other thing is that genetic diversity actually has real fitness consequences. So low genetic diversity can sometimes result in low fecundity or producing fewer offspring. And so high genetic diversity can mean that individuals have a higher fitness. And the other reason that I use genetics in my research is less about wanting to conserve genetic diversity as an element of biodiversity and more about how genetic markers can be used as a tool to help us uncover things about demography. So that's things like mating, reproduction, sex ratios, and dispersal. So we can use genetic markers to help us track patterns of relatedness, parent-offspring relationships, and things like that, and find out things that are really hard to find from just direct field observations, because the genetic data doesn't lie, whereas sometimes animals can be in, say, socially monogamous relationships, but there'll still be extra pair mating and things like that. So genetics is really great for just finding out exactly who's related to who and finding out those demographic trends. The other thing that genetic data can help us understand is how organisms interact with their landscape. So we can look at these patterns of genetic structure to figure out whether there are particular elements of the landscape that facilitate or block dispersal. So in this example, you can see there's a road, and individuals on one side of the road are more genetically related, they've got the similar colours, and on the other side of the road they're related, but the two groups are not related to each other. And that might tell us that animals aren't actually able to disperse across this road and mate with each other. And so, genetics can be a really great tool for helping us understand these landscape patterns and then feed this information through to conservation strategies. So in this case, you might put something like an overpass or an underpass for animals to disperse over. So this brings us to the first study I wanted to share with you, which is about using genetics to understand demography in the agile Antechinus, or Antechinus agilis. So this species is a small carnivorous marsupial in the Dasyuridae family. It's pretty abundant in southeastern Australia, and like many of the dasyurids, it has some really interesting life history traits. So these include things like torpor, which is it can go into daily to reduce its metabolic rate and conserve energy. it also has a really, like many of the antikynuses, it has a really short and predictable breeding season. So it breeds in the same week of every year, and it can live in groups of up to 20 individuals. The main goal of this project was to figure out if we can use different genetic marker types to learn more about dispersal and mating systems. And the reason it's important to develop new tools to answer these questions is because there are many assumptions held in ecology that actually do need to be tested and questioned. For example, usually we assume that mammals are polygynous and there's male bias dispersal. So that means that one male is mating with multiple females, and the male tends to be the one that moves further afield and establishes a territory after weaning, whereas females will settle closer to home, which is also called natal philopatry. But this assumption tends to overlook smaller, inconspicuous species where dispersal and social behaviours occur over a much finer scale. So to demonstrate this, I've grabbed a few examples from around the world with small mammals, where you can see there's a heap of variation. So we have different dispersal strategies, from restricted dispersal in both sexes in the American red squirrel through to high dispersal in both sexes in the greater spear-nosed bat, and a whole lot of variation in between, and then many different mating systems as well. And so if we can figure out how genetic structure at these different marker types is influenced by these processes, then maybe we can use genetic analyses to understand this complexity. The Agile Antikynus is a really excellent model system to test new genetic tools for understanding demography because number one, it's got a long history of being a model species in ecology, so it's been studied a lot. We know a lot about it. We know it has male bias dispersal, so the males tend to disperse roughly 500 metres, whereas females have really restricted dispersal to about 100 metres. And we know it has a promiscuous mating system, so both sexes are mating multiply. And so the different genetic markers we wanted to test were the mitochondria and the Y chromosome, and we use simulations to do this. And the reason that simulations are a really powerful tool for these sorts of questions is because we can parameterize them with a real species, so with the Agile Antikynus, with all of this excellent demographic data that we have. And then we can simulate different scenarios, so different dispersal capacities, different mating systems, and see how that affects structure at genetic markers. And the reason these two marker types are really useful for these types of questions is because whereas our autosomal markers are inherited by both parents, Y chromosomes are only inherited to males from their dad, whereas the mitochondria is inherited from your mother. And so this means that we can start to use sex-specific markers to understand sex-specific dispersal and mating patterns. The other reason that antikynus are really excellent for simulations is because they go through this thing called semal parity, which is adult die-off in the population. And so it's actually quite difficult to simulate multiple generations. And this means that generations are discrete, because after mating, you can see here from this sad-looking image that the male is not looking too good. And that's because they go so hard that often they'll have complete immune shutdown and internal organ failure, and they go so hard that one breeding season is it for them. And this is actually a somewhat common strategy across dasyurids, where individuals will put so much effort into mating that they might only make it for 1 or 2 breeding seasons. So it's been seen across the different antikynus species, and quolls, phascogales, and coludas as well. Another interesting aside is that even though some of the other dasyurids don't show semal parity, like the Tasmanian devil, it's actually been found that they've been able to switch from multiple breeding seasons to this semelparous strategy in response to the shorter lifespan that's been caused by the devil facial tumour disease. So we ran a whole lot of simulations to find out whether these different genetic marker types can help us understand demographic patterns in the agile antechinus. And what we've got here is some of the different strategies that we ran. So females are represented in with the circles, whereas males are represented by squares. And then we have the different Y chromosome variants or haplotypes represented in grayscale, whereas the different mitochondrial variants are represented in the red, yellow and orange colours. Our simulations showed that by comparing these different marker types, we can learn about the specific mating system. So for example, if we have a promiscuous mating system where 3 different males are mating with 3 different females within a litter, you can see that because the mitochondria is inherited from mother to daughter, all daughters in a litter are related. They have the same mitochondrial marker. And this is different across litters because they have different mothers. Whereas because litters can have multiple sires, multiple dads, you can see here that a different Y chromosome has been inherited from each father within each litter. And so males are not related within the litter. So if we see this pattern, we can be pretty sure that a promiscuous mating system is present. Alternatively, if there's monogamy happening where each pair only mates with each other, you can see that all males in a litter are related, all females in a litter are related, and this is, and across litters there's no relatives. There's no sharing of Y chromosomes or mitochondria. And then finally, in a polygynous mating system, we have a different pattern again. At first it might look a bit similar to monogamy, where all females are related to each other within a litter, all males are related to each other or share the same Y chromosome within a litter. But because this male has mated with 2 different females, you can see that across litters we have sharing of the Y chromosome. And so when this pattern is present, we can be pretty sure there's a polygynous mating system. These different marker types can also help us learn about dispersal. For example, if we just look at this situation where females are only dispersing 100 metres, whereas males are dispersing 500 metres, we would expect females from the same litter to end up being spatially close together, so they're more related to surrounding females, the red, orange and yellow circles, whereas males will be really unrelated because they've moved quite far and they're all mixed up. So we can use these different patterns of relatedness to pinpoint that male-biased dispersal is happening. Alright, so let's move on from simulations and get stuck into some real data. So the next study that I'm talking about is addressing this question that I mentioned earlier, where we can use genetics to understand how animals interact with their landscape. And for this research, we focused on the northern quoll. So the northern quoll is also in the Dasyuridae family, and it's an endangered marsupial that's declined across the majority of its range across northern Australia. There's a population in the Pilbara region of Western Australia, which is genetically and geographically discrete from the rest of its range. The Pilbara is a stronghold for the species because there's a lack of cane toads, which are known to cause mortality in this species because northern quolls will eat them and then die because of the toxin. Hopefully many of you are familiar with the Pilbara region, and some of you might have gotten to go up and see this incredible, spectacular landscape. But if you haven't heard of the Pilbara, it's a really vast and remote region, and it's an ancient landscape with one of the oldest geologies on Earth. The geology is so distinctive that you can actually see the Pilbara Craton from space. And although it's the size of a European country, it's got a fraction of the population density. It's also a really dynamic ecosystem, and it's very disturbance prone. So It can go from fires to flooding within the space of a year. And it's a multi-use landscape with a lot of mining happening, lots of agriculture, particularly livestock, and of course it's a tourist destination and a really culturally rich region, which is home to more than 30 Indigenous cultural groups. The Pilbara is also really well known as a biodiversity hotspot with high levels of species endemism. And so what this means is that there are many species that only occur in the Pilbara and nowhere else in the world. And this means that it's got very high conservation value. It's especially important for mammals, this conservation value, because Australia currently has the highest rate of mammal extinctions in the world. And you can see in this picture many of the species that we've lost over the last 200 years since colonisation have been marsupials. And so, as I was saying, the Pilbara is one of the last strongholds for the northern quolls because of its lack of this invasive species, the cane toad. But it's a mining and agricultural hotspot, and also many of the other threats that affect northern quolls in the rest of their range, like habitat loss, inappropriate fire regimes, predation by feral cats, and grazing by introduced herbivores, are all present in the Pilbara as well. And so if we want to sustain healthy populations of northern quolls in the Pilbara, then we really need landscape scale approaches to conservation. So ways to conserve the species, looking at the entire Pilbara as a whole, rather than just looking at specific populations or specific tenements in the Pilbara. But how do we do this? We need to maintain healthy metapopulations across the entire region. And to do this, we have to facilitate the mechanisms that support survival, which is these demographic patterns that we were talking about earlier, the ability to reproduce, to disperse, and of course to adapt to changing climate. So like the agile antechinus, the northern quoll also is an excellent model species for testing questions about how animals interact with their landscape and developing broad landscape scale modelling tools for conservation. And that's because there's been a heap of research on northern quolls over the last few decades. And what this means is that we know a lot about northern quolls and we have a lot of data floating around. So because there's so much research and data on northern quolls, we really wanted to utilise this existing data because conservation dollars are usually pretty limited. We used genetic data, so this is where previous projects over the last 20 years have captured northern quolls and taken a little ear notch. We can extract the DNA from that and have genetic samples for those individuals. And then we also used occurrence records. So these are records that have been uploaded to a database like the Atlas of Living Australia, where people have seen a quoll and then they record the coordinate and upload it to this database for us to use. And this can be ecologists doing surveys in the region, or it can just be any citizen scientist wanting to upload some of this data. And then we also gathered some spatial data to describe the Pilbara environment. So this was layers describing threats like fire and grazing intensity, climate, so rainfall patterns and temperature, terrain, elevation, which included elevation and ruggedness or rockiness and slope, geology, so the bedrock, vegetation, including the density of spinifex or woody cover like trees, soil, which included rock, clay, sand and silt, and also the location of more permanent water sources. And then we generated hypotheses about how and why the spatial data might be linked to where quolls occur and patterns of genetic diversity. So we used 4 main modelling approaches to figure out how northern quolls interact with the landscape, or how the landscape affects these really important life history processes. So the first one was using species distribution modelling to look at habitat, so what's important habitat for northern quolls. Then we used a method called genetic clustering to figure out if there were any barriers to dispersal. We used landscape connectivity modelling to figure out if there are particular corridors that act as that funnel dispersal, and then something called generalised dissimilarity modelling to figure out if there's any local adaptation happening to different environmental conditions. So I'm going to step through each of these modelling processes, so don't worry if that's a bit much for the moment. So the first method we use is something called species distribution modelling, which is the way that we find core habitat for different species. And so let me talk you through how this analysis works. So imagine we have this hypothetical landscape with spinifex grasslands, river, sand plains and rocky areas. And we capture this hypothetical rodent only in the spinifex patches. We can convert these captures to occurrence records by noting down their coordinates, and then we would upload it to one of these databases like Atlas of Living Australia. And then we convert the landscape into some kind of spatial data where each pixel describes an element of the landscape. So here you can see that the rocky, spinifex, sandy and river areas are represented by this image. We then use species distribution modelling to figure out which landscape attributes best predict our occurrence records. So in this case, the rodents were captured in spinifex, and you can see that spinifex areas have high habitat suitability. The modelers predicted that there's high habitat suitability in those areas. And you can also see that this information has been extrapolated outside of where we had those actual captures. So anywhere with spinifex, we can say it is probably good habitat for the rodent. So we used this approach for northern quolls, and we came up with a few predictions to help us decide which spatial layers to test. So we predicted that habitat complexity, so whether this be terrain ruggedness and rocky areas, or whether this be about habitat complexity or habitat vegetation density, we predicted that these things would provide protection to northern quolls from predators while they're mate searching, denning and foraging. So for pups in dens as well. We also predicted that fire and grazing would have a negative impact on habitat because they remove habitat complexity provided by plants. And then we thought that the climate would shape where northern quolls are found, but not so much through kind of a direct relationship, because we know northern quolls are pretty good at sheltering from extreme climate conditions because they do have these dens in rocky areas that are buffered from temperature. But we thought the climate might shape where their prey species are found. So here you can see our species distribution model that we ran for quolls, and it's a map of the Pilbara on the right, where the darker areas have low habitat suitability, so there probably aren't any quolls living in those areas, whereas the lighter yellow areas have really high habitat suitability. So they represent really core habitat for the northern quoll. And the landscape elements that are common in these areas with high habitat suitability are terrain ruggedness. So there's high terrain ruggedness, there's high elevation, and they're close to water, close to more permanent water sources. So these are the things that are really important for driving key habitat for northern quolls. So some of our predictions seemed to be right. Habitat complexity is important for predicting where quolls live, and this is probably linked to some kind of predator avoidance or protection from predation. And we've also seen these findings in other studies before as well, which are listed below, so it's a pretty robust finding. Next, we use something called a genetic clustering algorithm, which I'll describe in a second, but this method we can use to find out if there are any barriers to dispersal in the Pilbara for northern quolls. And so we had a few predictions for this as well. We thought that aridity might act as a barrier to dispersal, so aridity would be more representative of the deserts, and we know that the Pilbara is really genetically unique and separated from the rest of the northern quoll population, and it's surrounded by deserts, so it could be that desert is acting as a barrier. It's quite hard for northern quolls to move across this habitat type. And we also thought that rivers might act as a barrier to dispersal because they're difficult to cross if they've got high water flows. If we want to look for barriers to dispersal, we can use this genetic clustering algorithm that I mentioned. And the way that this works is if we have these populations of rodents, and I'm sorry that they're rodents when this is a marsupial course, you can see what my main research focused on before, but anyway, let's call it our hypothetical rodent species. And if there's absolutely no dispersal between these different populations, then you would imagine that these three populations would represent three genetically distinct clusters. So individuals would be more genetically similar to other individuals in their population than to individuals in the other two populations. On the other hand, if there's really high levels of dispersal between these three populations, then we'd expect them to be genetically mixed. So individuals could be quite related to individuals in other populations. When we ran this clustering analysis for northern quolls, we didn't find much evidence for really strong barriers to dispersal in the Pilbara. And you can see that by looking at this structure bar chart up the top here, where each bar represents an individual and the colours represent the proportion that they align genetically to the two genetic clusters that we detected. So here you can see some individuals are mostly purple, which means that they're mostly genetically similar to the cluster number 1. Some are mostly pinky red colour, which means that they mostly align to cluster number 2. But there's this real gradient between them, meaning that there's probably quite a bit of dispersal between the two populations. And so what this might mean is, even though there's probably no contemporary barriers to dispersal, there might have been a historical one. So the Yule River is about here in the Pilbara, and this area dissecting the Pilbara is the Fortescue Marsh. And if we go back about 15 to 25 million years ago to the mid to late Pleistocene, we know that this is the period where aridification started to happen. So these rivers were probably a lot stronger about that time, and then slowly as the Pilbara started to become more arid, they've probably become more easier to traverse. And so we think this pattern is probably more historical than contemporary. So just because we didn't find any really hard barriers to dispersal for northern quolls, it doesn't mean that some habitat types aren't easier to move through or more difficult to move through for this species. And so to find this out, we used landscape connectivity modelling. And again, we made some predictions about which areas we thought would facilitate or impede dispersal. And again, we thought that ruggedness and complexity would be the most important habitat types for providing protection to northern quolls from predators while they're dispersing. And because they're semi-arboreal, so they're quite good at moving through or traversing rocky habitats, we thought there might be some biomechanical constraints for moving through particular substrates as well. So maybe sandy substrates are harder for them to move through. So if we go back to our hypothetical landscape, you'll remember last time we identified our important habitat patches. So now we want to know how animals move between these patches using the connectivity modelling. So if we're back at this landscape and we've caught our hypothetical rodent species, we then get genetic information from each individual. And parents pass this information on to their offspring because genetic information is heritable. And over multiple generations, as offspring disperse, we start to see patterns of relatedness build up across the landscape that tells us something about connectivity. So some parts of the landscape might offer an advantage to dispersal. So we predicted that maybe rocky areas provide protection from predators, whereas other areas are more difficult to move through and animals don't survive if they try to pass through maybe sandy patches or rivers. And so this means that individuals that are connected by rocky habitat are more genetically similar, whereas areas separated by a barrier like a river are genetically distinct. So we can create something called a resistance surface that reflects this information. For example, here we've got our brown rocky pixels, which are given a really low value, a 1, to show that they're easy to move through, whereas rivers get a really high value to show that they're difficult to move through. So we've given them a 100 here. And then we imagine that we're pumping an electrical current through this resistance surface. And this brings up the areas that act as dispersal corridors with really high connectivity, and areas that are kind of acting as barriers or difficult to move through, like the rivers, and they get a really low value for connectivity. And in this way, we can start to visualize these genetic patterns across the landscape. So when we ran this analysis for northern quolls, we found that unlike our species distribution model, where terrain ruggedness or those kind of rocky, rugged areas were really important for acting as core habitat, actually terrain ruggedness wasn't at all important for dispersal. So here you can see our connectivity map of the Pilbara, where the lighter colours represent areas that are easy to move through and the darker areas represent places that are difficult to move through. And the first thing you might notice is that there's pretty high connectivity across the Pilbara in general, which is good news. However, we did find that areas close to water or permanent water sources acted as dispersal corridors. So that's some of these trails you can see. And then we also found that the areas that were really difficult to disperse through tended to have high silt content. So that's usually these alluvial and hardpan plains, these really bare areas that probably have very little protection from predators. The other thing we found is that, so here the circles represent genetic diversity of the individuals in that area, and you can see that we had much higher genetic diversity in areas with high connectivity. So because there's lots of dispersal, there's lots of genetic exchange, which is really important for conserving genetic diversity. But then in areas that were either separated by a barrier, so here this is Dolphin Island, which of course is separated from the mainland by the ocean, or areas that didn't have a lot of connectivity. So here you can see the Fortescue Marsh is acting as a pretty strong barrier to dispersal, and there's only a few dispersal corridors or weak dispersal corridors across the Fortescue, and we have much lower genetic diversity in those areas. And so this might give us a little bit of an indication of areas we really need to focus management on and make sure that we don't start seeing really high levels of inbreeding and, I guess, eventual population decline in those places. So our final question was whether different populations of northern quolls in the Pilbara might be locally adapted to different environmental conditions. So our main prediction here was that local adaptation might be driven by climate, but this would be kind of indirect because climate might influence food resources. So the different kind of prey items or fruits that are available, fruit and vegetation that are available for quolls to eat in different areas. And this would be really important because it could influence their reproductive success, because lactation really increases the energy requirements northern quolls need. So maybe in areas that have food resources with low energy content, they might be reproductively less successful than quolls in other areas that have really high quality food resources. The way we do this is to look for signatures of local adaptation to specific environmental variables in the Pilbara using something called generalised dissimilarity modelling. And what this means is that we're looking for patterns of genetic differences that are linked to environmental gradients, and we call this genetic turnover. So we're back at our hypothetical landscape, and we've got these individuals where there seems to be kind of two genetically different groupings of individuals. However, in this case, the genetic patterns don't seem to link up with any of the physical landscape attributes. So not with rocky areas or with spinifex or sand or anything like that. But instead we see that genetic differences are linked to this different climate gradient. So perhaps we have a warmer climate in the west and a cooler climate in the east, and we can see that individuals are locally adapted to these two different climates. And so there's genetic similarity within similar climates and genetic dissimilarity across different climates. So for northern quolls, we did find some evidence that genetic turnover was driven by different climates, specifically by temperature extremes and rainfall seasonality, which suggests there might be a little bit of local adaptation based on climate. However, these patterns were really subtle, and probably because of the high levels of gene flow or dispersal in the Pilbara, it's unlikely that there's really strong local adaptation to these different climates. So you can see here we've got our map of the Pilbara, and the colours represent these different genetic gradients. So individuals in the regions that are similar colours are genetically similar, whereas if they're in regions with different colours, then they're likely to be genetically different. So just to sum up, we found that the landscape elements that were important for core habitat or for connectivity or dispersal were really quite different, with habitat being predicted by terrain ruggedness, whereas connectivity was predicted by distance to water or proximity to water. We didn't really find major barriers to dispersal, but we did find that alluvial and hardpan plains were difficult to move through for northern quolls. And we found that climate played a really subtle role in driving genetic turnover, so there wasn't very strong evidence that there's local adaptation, which is good news in terms of changing climates in the future. Northern quolls are probably quite capable of surviving in many different environments. So by understanding how northern quolls use the landscape and how this links to their underlying ecology and demography, we can funnel this information into regional conservation for the species. So the Northern Quoll Project is a really nice case study for developing some of these landscape scale conservation tools. But to finish, I wanted to tell you about how we're expanding this analysis to the entire small mammal community in the Pilbara, but specifically for the marsupials in the region. So along with northern quolls, we also have genetic data for 6 other marsupial species in the Pilbara. And then we also grabbed the species occurrence records for 11 additional marsupial species as well across the Pilbara. And then we ran the same analyses across these species as we did for northern quolls. And so for looking at core habitat across marsupials, we found that there was a really mixed response, which was mainly based on terrain and soil. So for example, where northern quolls really relied on rocky habitat, bilbies require really sandy areas, whereas things like the Pilbara Ningawi are really intermediate between these two extremes, so they're more generalist. There was also usually a preference for high aridity or high precipitation. So again, a real mixed response, either being very well adapted to aridity or arid conditions, or being more adapted to that kind of monsoonal tropical climate. And this probably reflects that there's more of a fixed link to specific environments in the marsupials because they've actually adapted physiologically to these conditions. So things like fat storage and torpor can really mean that they can adapt to a specific environment, and so climate is a really strong predictor of where they occur. So in terms of connectivity, we found that rocky areas tended to act as barriers for many of the marsupial species. So again, you can see that the Fortescue Marsh here is acting as a real dispersal corridor across many marsupials, and a way to kind of get into the rocky areas in the Pilbara. And these results really contrast with our habitat models, showing that different elements of the landscape can be really important for different ecological processes. And so we're feeding all of this information into a suite of decision-making tools for conservation in the Pilbara, including species richness maps, where we stack that habitat layer across multiple species to find areas with high species richness versus areas with low species richness. And we stack these connectivity maps to find multi-species dispersal corridors in the Pilbara. These will be really important for targeting management activities and for looking for important areas to protect across multiple marsupial species. So that's it from me. Thanks for sticking around to hear about how we can learn about marsupial ecology through genetics. And I just wanted to finish by thanking funders and all the wonderful collaborators that have worked on these projects with me.